The topic of my talk is actually well situated in regards in the program because I'm going to be talking about the GI tract and in particular the millions and millions of bacteria that live in the gut and how important they are for our health, and in particular the emerging concept that they're involved in lots of chronic inflammatory diseases, particularly those in which there's an immune cell involvement. So these billions and billions of bacteria are busily now digesting the food that you've just been consuming. So I hope at the end of my presentation I'll have a much more appreciative opinion of their role in health. So as Ian said, I'm presenting this on behalf of both myself and Tom Wildman. We're both members of the UEA. Tom is based at the university. I'm based at the Institute of Food Research, but we're both UEA employees. So I'll just give one slide on the GI tract, just as a refresher, and then I'll go into the microbes in the GI tract, which we collectively call the microbiota, talk about their importance in health and their emerging concept of their involvement in triggering chronic inflammatory diseases, and then something about what the Norwich Research Park is and what we think we can bring to this area of research. So the GI tract, as you all, I'm sure, can appreciate, it's the inner tube of life, and it plays an essential role in digestion and in particular extracting nutrients from the things that we consume. It's a 9-meter long tube, and amazingly it's just lined by a single very, very thin layer of epithelial cells. And what's often not appreciated about the gut is in fact that it's the largest immune system in the body. And it also has the second largest number of neurons. And the enteric nervous system controls virtually most of the vital functions of the GI tract. So when people say they get a gut feeling, there's an awful lot in that. It's your second brain talking to you. And also importantly, from the perspective of infection and disease, it's the major route by which pathogens gain entry into the body. And in addition, it's in intimate contact here with a vast number of microbes and is exposed on a daily basis to lots and lots of things that we consume, the food. So it has numerous challenges. And in this slide, I've just tried to illustrate the important decisions that the gut has to make on a daily basis. So as I said, it's a single layer of epithelium that protects us from what's in the lumen of the gut. It gets exposed to things like this on a daily basis, so our food and also our resident bacteria, microbes. And normally, we don't respond to them. If we did, we'd have food hypersensitivity and we'd have some awful autoimmune disease. However, we also come into contact with the nasty microbes in the environment. So these can be parasites, viruses, pathogenic bacteria that's present in our food, salmonella being a good example. And the immune system here in the gut has to mount a response to them, otherwise we get sick and we get ill. And so it really has to be able to discriminate between what's good for us and what's bad for us. And hopefully what's bad for us is acted upon here in the gut and it leads to eradication, containment. If that fails, then we end up with disease. Importantly now, what's an emerging concept is that a breakdown in tolerance, so in other words, we no longer ignore the good guys, but we start to see them as bad guys, can lead to quite severe disease. And one of the most extensively characterized diseases which is linked to this breakdown of tolerance is inflammatory bowel disease. So a little bit more about what the microbiota is. So bacteria line the entire length of our GI tract, from the mouth to the anus. And what this slide illustrates is as we go from the top to the bottom, there's an increasing number of microbes that reside in our GI tract. So there are a small number in the mouth, oral cavity, slightly more in the stomach, where they have to survive the acidic challenge that the stomach poses. Small intestine, increasing numbers, until we get to the colon, the large bowel, where we have phenomenal number of microbes. This is 10 to the 12. This is trillions and trillions of bacteria that essentially fill up all the space that we have in our colon. Collectively, there are about 10 times as many bacterial cells as we have cells in our body. So we're essentially outnumbered by these microbes. And if we start counting and collecting the genes that these bacteria have, they outnumber us by about 100-fold. So in terms of our genome and our makeup, there are two components to it: the genes that we have in our own cells and the genes and the activity they encode in the bacteria that live in and on our body. And that's often not really appreciated. So up until about 10 years ago, the only way we could start to determine what's in the gut and what they're made up of was basically taking them out from stool samples and trying to culture and grow them in the lab. And that was relatively successful, but it was just really the tip of the iceberg. And then about 10 years ago, along came some of the omics technologies you've been hearing about, so genomics, whereby we could identify bacteria based on unique genetic identifiers that reside within their genomes. And this culture-independent profiling is basically based on extracting DNA from these vast number of microbes, generating the ribosomal DNA fragments by a PCR-based reaction, and then sequencing them through massive machines. And we end up with being able to identify the genes that correlate to the bacteria. And so this has led to an explosion of interest, and we're now cataloguing all the types of bacteria that live in our GI tract. And this has led to the discipline of metagenomics. This is another omics technology. And basically, it allows us to take the sequence information that we have here and translate that to function. So, because most of the genes in bacteria code proteins that have a useful function, about more than 80% of them. So based on the sequence, we can predict what that means in terms of proteins, enzymes, etc., that these bacteria make. And we know from this that there's a significant number of genes that are unique to the bacteria in our gut. And not surprisingly, these are related to the breakdown of food material from which we benefit by extracting nutrients from. So as a result of this, several groups have looked at the relatedness or similarity of the microbiota in the GI tract of different animals. And they've come up with this, what's called a network analysis. This is a map of relatedness. And so if we put humans and primates on this, we all cluster around here, which is probably not surprising. If we look at lions and bears, these carnivorous animals, they're slightly off this, so they're distant from, but somewhat related to the bugs that we have in our GI tract. Horses and rhinos, different type of diet, different types of animals, they're up here. And then we have the sheep, the cows, the ruminants down here. And some more exotic species, so elephants are up here, more related to horses and rhinos. And then what's interesting here is we've got leaf-eating monkeys, which are separate from the carnivorous primates. And so what this means is that phylogeny and the diet shape the types of microbes that we have in our GI tract. And if we go further and look at, well, what types of bacteria are present in our gut, what this shows is that in different environments, so this is water, soil, and salt water, freshwater, salt water, the different colours represent lots of diversity, so lots and lots of different types of species. We come down here to our own guts, and you see just two colours primarily, red and blue. So two species make up the vast majority of the bacteria that reside in our GI tract. And that distinguishes us from all these other environments. So for some reason, we've undergone an evolutionary process that's selected for very small numbers of dominant phyla in our bacteria. And not surprisingly, there's lots of large-scale projects going on now to define what the human microbiota is. And groups in Europe and the US, this is the summation of their knowledge to date. And they've identified what they believe to be a core intestinal microbiota in humans. And from these studies, they've said the microbiota is highly variable between individuals. Interestingly, though, family members share similarities in microbiota more so than unrelated individuals. And twins are more related still than other siblings. And what's key in terms of developing the microbiota and how these bacteria start to populate our gut is the environmental exposure we have, and I'll talk a little bit more about that. But what's come out of the sequencing program is that there are 75 species that are present in more than 50% of us. And breaking that down further, there are 57 that are found in virtually all of us. And these cluster into just 3 types of organisms called enterotypes. And these dominate worldwide. So independent of sex, gender, race, geographical location, these are consistently found. And the 2 phyla that predominate, which were the red and the blue stripes in the previous graph, are this one's here, Bacteroides and Firmicutes. So those 2 predominate in all of us. And this functional core is highly conserved in everyone. So these bacteria are present because they're involved in this process which is essential for gut health. So they will degrade complex carbohydrates, so plant material that's part of our diet. Without these bacteria, we couldn't break that down. Synthesize short-chain fatty acids, which are products of this degradation of complex carbohydrates. They produce indispensable amino acids, which are the building blocks of our proteins, which form our tissues and organs. And they produce vitamins that we cannot get from any other source. So vitamin B12, for example, comes from the bacteria that live in our GI tract. Just recently, this analysis has been extended to viruses, and we now know a lot more about the viruses that populate our gut. And this was published last year, and the results here are summarized here. So there's a mixture of viruses of eukaryotic origin, and also phage and prophage genome sequences have been identified. Co-twins and mother share significantly greater degree of similarity than unrelated individuals, so that's similar to the bacteria that are present in the GI tract. And these viromes are unique to individuals, so each of us have a distinct population of viruses, and depending on how related are to other individuals, we may share some or none of those. And what's interesting is the interpersonal diversity is low, which means once we have them, they stay with us, probably throughout our lifetime. This study was restricted to 1 year period, but it seems likely that they are with us from birth to death. And their functions are predicted to be useful indicators of health and also perhaps impending or fully manifest disease states when these viruses presumably replicate, reproduce, infect some tissues, and maybe cause disease. So viruses are becoming increasingly more of interest to people that are characterizing the microorganisms that live in our GI tract. So, as I said, the microbiota is essential for our health and well-being, and these are some of the core benefits that they provide to us. They populate all the space in our GI tract, which means they can restrict colonization by pathogens, which may come through food and water that we consume. They're an essential source of vitamins and other cofactors that we need for healthy metabolism. And they provide about 15 to 20% of our daily energy requirements through their production of short-chain fatty acids, which they generate from the breakdown of plant material that's part of our diet. And they also promote or maintain the epithelial barrier, this single layer, this less than paper-thin layer of cells, which protects us from what's in the gut. And they also promote peristalsis and motility, and local immunity as well. So in the absence of microbes in the GI tract, our immune system is severely compromised. So there's an intimate relationship between the microbes in the gut and our immune system. And this relates to oral tolerance, as I said in one of my first slides, the ability to distinguish what's good for us and what's bad for us. So we know from experiments done with animals, which we can raise and maintain as sterile animals, so these are maintained in a bubble, so they never get exposed to any microbes or whatever, so they are sterile animals. These animals are profoundly different from those that have microbes that populate their surfaces and internal organs. So they have a defective epithelial barrier. They have a leaky gut barrier. And they also have defective lymphoid tissue development, so their immune system is poorly developed. And this leads to a defective immune response. And in particular, the production of IgA antibody, which is one of the primary mechanisms of protection and defense in the GI tract. And this results, not surprisingly, in increased susceptibility to infection. So these animals have to be kept sterile and protected from pathogens, otherwise they would probably die from infections. So what's this got to do with the brain and the CNS? Well, there's quite a lot of evidence, some of it anecdotal, some of it much more substantive, that links the gut and the microbiota and the brain. And it's been known for a while that there's a beneficial effect of oral antibiotics and laxatives on certain types of encephalopathies. This one is hepatic. Acute infection is often linked to anxiety, depression, and cognitive dysfunction. And to some extent, introduction of beneficial microbes, so-called probiotic microbes, can improve this to some extent. And intestinal infections negatively impact on memory and learning. Again, there's some evidence that administering beneficial microbes can prevent or limit this impact on CNS. Now, this is an interesting paper I picked up from last year by a group at the Karolinska Institute, where they looked more directly at the effect the microbes in the gut has on development and behavior. And this sort of in italics here is a summation of the results of this paper, in which they showed the microbiota can have a significant and profound impact on brain development, but also on behavior functions. And this is by modulating the production and response to chemical transmitters in the brain that affect motor control and anxiety or fear extinction behavior in adult life. And so these could all be restored or repaired by introducing right types of bacteria into these animals. So this really sort of substantiates this emerging concept of the gut-brain axis. So here we have the ability of the brain to influence the gut, and in particular the microbes in the gut. And this is shown here via generally stress-induced changes that leads to changes in the GI tract, so in the physiology, epithelial function, which you can translate to barrier function, mucin production, epithelial cell function and motility, which in turn releases neurotransmitters by the enteric nerves, and this feeds then back into the brain, and so we have this loop, communicative loop between the gut and the brain. And on the other hand here, it's now emerging that the microbiota can impact on the central nervous system through the activation of neural afferent circuits to the brain, activation of mucosal immune responses, and release of transmitters which can impact on CNS activity, and metabolites themselves, so what the bacteria produce in the gut, these can, once they enter the bloodstream, can directly influence the CNS and behavior in general. So what factors impact on the establishment of the microbiota in the first place? And there are 4 categories of effectors or inputs that shape the bugs that we have and that we live with for the rest of our lives. First one, not surprisingly, is genetics. And we know this because of mutations that have been identified in certain genes, which seem to have a profound effect on the relationship between the microbiota and our immune system. So these genes here are all identified as susceptibility genes for developing Crohn's disease, a type of inflammatory bowel disease. And we now know that the change in the microbiota trigger these inflammatory bowel diseases. Lifestyle is important, so the diet, stress and activity impact on shaping the microbiota. And birth and nurturing are very, very important. So whether or not an infant is born in the hospital or outside a hospital will mean they get exposed to different types of colonizing microbes. Vaginal versus cesarean delivery has an impact on the first type of bacteria that invade the body. So vaginal birth would mean the mother is providing a source of microbes that will inhabit the infant, whereas cesarean section, that generally doesn't happen. And then bottle versus breastfeeding also promotes the growth, colonization of different types of bacteria. And then, not surprisingly, medical practices also have a significant impact through antibiotics, hygiene, and vaccinations. And what these factors are inputting in is to the balance here between the symbionts, which are our beneficial microbes, and the pathobionts. And these are the potentially disease-causing bacteria that reside in our microbiota. If this pathway is stimulated, then this can lead to inflammation. And this is where pathogens have a major impact on gut health. They're tipping the balance in this direction. Ordinarily, and hopefully most of us, this dominates here, so we've got regulation and homeostasis. So just a couple of examples of how these factors can impact on the microbiota. The first one relates to how diet influences our microbiota. And these experiments relate to animals. So there are strains of mice which are morbidly obese here. And they have alterations in these 2 dominant populations of microbes I talked about that make up the microbiota in most vertebrates. Here we have an increase in firmicutes and a decrease in Bacteroides. Putting these animals on a diet results in them losing weight, and we get a change in both firmicutes, which now decrease, and Bacteroides now which increase. So an association of diet impacting on the microbes, which impacts on physiology. If we go the other direction until we take a lean mouse and feed it hamburgers, then within 8 weeks it starts to become morbidly obese. And again, we're detecting changes in the microbiota. So there's a decrease in diversity, and again this increase in the firmicutes. And they, not surprisingly, gain weight, and they have lots of more fat deposits. And then if we put these fat mice on a reduced diet, so we take away a lot of the fat and the carbohydrate, they end up becoming lean mice. And this is related to a decreased calorie intake, decreased weight gain, and an increase in the bacteroides. So what we eat impacts on our microbiota, which in turn relates to how much energy they're producing and how much energy is available for our uptake. If that's in surplus, we gain weight and become obese. The second example is how gene mutations can impact on the microbiota. And these, again, are experiments in animals. And what we have here are wild-type animals, and animals that are genetically identical with the exception they have a mutation in one single gene. And what we've done here is used a technique to profile the ribosomal components of the bacterial species that these animals have in their GI tract. So the bands represent different populations of microbes. So you can see in these three animals here, there's lots of, as we'd expect, lots of similarities. When we look at the mutant animals, we can see there's huge differences. So one gene mutation, these animals are raised in the same cage, same diet, same environment, yet one gene mutation has a profound impact on the microbes that reside in their gut. Using metabolomic analysis, where we're looking at the metabolism of the microbes in the GI tract by NMR and mass spec, you can see here the mutant mice in blue and the ones in green. You can see there are different peaks in the blue profiles than in the green. And this translates to metabolites which are implicated in bile salt synthesis and fat metabolism. Interestingly, these two different mice are profoundly different in their ability to resist infection by Salmonella, a major foodborne pathogen. So if we infect these mice with Salmonella and look at the numbers of bacteria in their tissues, the wild-type mice can deal with these quite effectively and at very low levels. The mutant mice with this altered microbiota are now profoundly susceptible to infection by this pathogen. So these changes in the microbiota result in changes in metabolism and what they produce, which impacts on their ability to resist GI tract infections. So that's two examples of that. So the microbiota and disease. And what we have is that we have the enemy within. So we have within our microbiota populations of microbes which, under the right conditions, can cause quite profound and severe disease. And here are some examples. The best one is Helicobacter pylori, which you heard about earlier relating to stomach cancer. It's part of our microbiota in about 25% of us. Fortunately, most of us are able to live with this and it never causes any problems. Enterococcus species and Clostridial species are also part of our microbiota. Clostridium difficile is a good one that you may have heard about as infections in hospitals. These normally will be controlled and contained by the good members of the microbiota, but there are conditions under which these will flare up and be allowed to grow. Good example is antibiotic overadministration in hospitals, which depletes the good bacteria in our gut, allowing this particular one to expand and cause disease. And then host nutrition. I've told you that if our gut bacteria produce too much energy, we can take that up and it gets stored as fat. So it's not all good news. Interestingly now, there's lots of evidence to suggest that gut microbes play a role in development of autoimmune diseases. And this sort of table summarizes this. It's a variety of autoimmune diseases, some that affect the CNS. And you can show that if these susceptible animals are kept under germ-free conditions, so they never come into contact with microbes, or they're treated with antibiotics, generally there's a positive impact on the disease severity, and some animals, in fact, don't get disease. So there's a clear link between the microbes in our gut and the susceptibility we have to developing autoimmunity. And so I've scanned the literature to look how this may be related to CFS, and there are a couple of examples here. So many CFS patients have GI disturbances, particularly irritable bowel syndrome, and they may have a leaky gut barrier. If there's a leaky gut barrier, then that means our contents in our gut can get access to our body, and they can overstimulate the immune system. We have this chronic inflammatory condition. Some preliminary analysis has shown that the microbiota in CFS patients is different to that in normals, healthy controls. And in some, this can be attributed to a particular type of bacteria called lactic acid bacteria. And there's an increase in this bacteria. This can cause acidosis, increased acid production, and also high anaerobic enterococcal counts are correlated with more severe types of CNS disorders and cognitive defects. And then there are lower levels in some patients of bifidobacteria in CFS patients. So again, there may be some grounds here for further investigation. But then the question is, well, how do we identify what the good guys are and what the bad guys are? So it's rather like looking for Waldo. So I'll give you 2 seconds or 3 seconds to try and find Waldo. No? There he is. Okay. So this is the task that faces when we try to identify what may or may not be the bad guys among all the good guys. However, it's never that simple because, as I said, there's a potential for any good guy to become a bad guy. So we end up with lots of Waldos. And so trying to identify what the organism is that triggers all this disease process is incredibly complex. But we are starting to make significant inroads into addressing this question. And one good thing is that we do know, we've known for over a century, that we can manipulate the microbiota for promoting health benefits. And there's enormous potential here for lots of types of diseases by eliminating the bad guys and promoting the growth of the good guys. And really it all started with this guy, IIMEC6, who got a Nobel Prize in 1908. And what he observed was that Bulgarian and Russian peasants who lived largely on fermented milk seemed to live longer than those that had a different type of diet. So he introduced the first probiotic, which is sour milk. And this really underpins all the probiotic technology and the developments in the field that's happened since. And probiotics is essentially just giving good microorganisms to us that will promote health. And not surprisingly, lots of companies have sort of latched onto this, and there's now, it's $1 billion industry, and some of these you may have seen, or in fact some of them you may be taking right now. The bad news is that there's no scientific evidence to support the health claims. Okay? So don't bother unless you are really ill. However, clinically, when given to patients who actually have a disease, there is a good indication that these are beneficial. So in rotavirus infections, pouchitis, bladder cancer, these are all positive indicators of a beneficial effect of probiotics. And I think this is the way forward. Rather than health and sort of improving health, I think probiotics have a real role to play in treating disease of the GI tract and beyond. And there's one study I found in the literature where there's some evidence that this might be useful in CFS. This is a study done in which CFS patients were given lactobacilli. This is the Shiroti strain, this number of bugs, and it had a beneficial effect on anxiety symptoms, and it raised the level of the good guys in the GI tract. And the good guys is lactic acid bacteria of antioxidant properties, so that may be beneficial. That may be a mechanism by which they have these beneficial effects. So all of what I've told you is really something that we are very focused on and is becoming an area of expertise in the Norwich Research Park. For those of you not familiar with it, here we have the University of East Anglia, where Tom is working, the Norfolk and Norwich Hospital, the Institute of Food Research, where I'm based, but we also have 3 other institutes. And the one that's particularly of interest to us is the Genome Analysis Centre, which Ian mentioned earlier. And this is specialised in the sequencing of non-human genomes, which is dealt with by the Sanger Institute. So we are actively engaged with these people in sequencing microbial genomes, which I think is going to be something that really makes us distinctive and unique. There's no other centre like this in the UK. So the expertise we have isn't present anywhere else outside of Norwich. And this just exemplifies that. So at UEA, we've got virology that Tom leads on, GI tract immunology, which I contribute to. The Genome Centre is Genomics, and then the Institute of Food Research is GI tract microbiology, epithelial cell biology, and immunology. Put them all together, and that's a pretty powerful combination of scientific disciplines. And we are doing that. We are putting them together, and these are 3 areas that we've identified where Norwich can become world-leading in about the next 5 to 10 years. And gut biology in health, aging, and disease is front and centre of that enterprise. And I think that's what we can contribute to this area of research in particular. And on that note, I'll end. Thank you very much.